Good day everyone. So for this session, I am to discuss a little introduction about fungi. So we have here the introduction to mycology. For the learning objectives at the end of this lesson, the students are expected to discuss the characteristics of fungi, reframe the common terms in mycology, identify fungal structures and morphologic similarities, and differentiate sexual and asexual reproduction. So first, let's have the basic. So we have two general categories of microbes. So number one, cellular, and number two, acellular. So I have here a diagram showing the categories of microorganisms. So for acellular microorganisms, we have in particular the viruses. So they are acellular. And then for cellular microorganisms, we have prokaryotes and eukaryotes. So prokaryotic cells and eukaryotic cells. So one of the main differentiating points of eukaryotic cells and prokaryotic cells is that the eukaryotic cells, they have true nucleus which is enclosed in a membrane. So that is where we can find their genetic material. However, for prokaryotes, they don't have true nucleus. So the nucleus there is what we call as the nucleoid. So again, prokaryotes don't have true nucleus. What they have is a nucleoid. So that is where we can find their genetic material. So also we have here the examples. For prokaryotes, we have eubacteria, cyanobacteria such as the blue-green algae, and of course archaeans. So for archaeans, these are microorganisms that can live in an extreme environment. Like for example, the thermophils. So thermophils, they are microorganisms that can live or can tolerate very high temperature. So by the name itself, thermophils, they can tolerate extreme temperatures. We have another one, halophils. So meaning to say, these are microorganisms that can tolerate high salt concentration, or meaning to say, they can live in an environment wherein there is high salt concentration. And for archaeans, we also have methanogens. So they can use methane like as a source of their energy for their growth and for their differentiation. Okay, how about for eukaryotes? For eukaryotic cells, we have fungi, we have protozoans, we have algae, we have plants, and we have animals. So for this discussion, our main focus is, of course, the fungi. So what is mycology? Again, as what I have mentioned, mycology is the study of fungi. That comes from the Greek word mykos, that means mushroom. So the fungi class is present in the environment as saprobes. So they are saprophytic. So what do I mean by that one? So saprobes, meaning to say they rely on decaying matter, like decaying vegetation and plants for sources of nitrogen. So that is one of the characteristics of fungi. And then also, other than the word mycology, we have other terms related to this. So when we say mycologist, okay, these are persons or these are scientists, for example, who study fungi. So mycologist. When we say mycotoxicology, what is it? Mycotoxicology, it's the study of fungal toxins and their effects to the body. So that again, mycotoxicology. Okay? How about the diseases caused by fungi? So the diseases caused by fungi, okay, that is mycosis. Okay, mycosis. So the diseases caused by fungi, or the study of the diseases caused by fungi. Another thing, we have the distinguishing characteristics of fungi. So we have 5 main characteristics. So number 1 again, fungi are eukaryotic. And aside from that, they also are non-vascular. Number 2, they don't have chlorophyll, unlike the plants. And also, they are non-motile. Number 3, cell wall is made up of chitin. Number 4, obligate aerobes. So meaning to say they require oxygen to grow, to replicate. Okay, just like how you see the growth of a mushroom. Because again, mushroom is an example of a fungi. Okay, and fungi grow best at a neutral pH. And lastly, they need moist environment which are necessary for their growth. And take note, fungi contain spores. So that spores that they have can withstand even dry environment. And they also reproduce by means of their spores. And that spores, we call it conidia. Okay, one of the examples of spores, conidia. Okay, and also for reproduction, fungi can be having sexual reproduction, okay, in the process of meiosis, or they can also reproduce asexually, okay, like the mitotic process. So that's for the reproduction of the fungi. Again, fungi are eukaryotic. So meaning to say they have true nucleus that is enclosed by nuclear membrane. They may produce energy also, so that means they have the mitochondria, which is responsible for energy production. Because for prokaryotes, again, they don't have true nucleus and no nuclear membrane. They only have nucleoid. But bacteria can also produce energy, but they don't have mitochondria, right? So what they have are mesosomes. Mesosomes are a structure in prokaryotic cells that help them, okay, reproduce, okay, for cellular reproduction and energy production. Okay, those are again mesosomes. Next one, fungi don't have chlorophyll. So meaning to say they cannot synthesize their own food because again, as what I have mentioned, they are saprophytic. They are saprobes. So they get their food from soil, from water, from organic debris, or any decaying matter. Okay, but plants, okay, they have their own chlorophyll. So meaning to say they can synthesize their own foods. And also, another differentiating points between, of course, the fungi, like the animals, okay, and also the plants, okay, is the storage of their food, okay. So for the fungi, they store their food as glycogen, like in animals, okay. They store their food as glycogen, whereas the plants, they store their food as starch. And another thing, the cell wall of the fungi is made of chitin. So this chitin is responsible for very rigid cell wall of the fungi. So that means it gives shape to the fungi. How about for the plant cell wall? The plant cell wall is made up of cellulose. Okay, cellulose. Another thing, we have here the differentiating points between, again, prokaryotic cells like bacteria and eukaryotic cells like fungi. So again, cell type, prokaryotic, unicellular, fungi, eukaryotic, unicellular, or multicellular. For the roles in ecosystem, bacteria can be both producers and decomposers, but for fungi, they are saprobes, so they are mainly decomposers. Optimal pH, neutral for bacteria, slightly acidic for fungi, 4.0 to 6. Cell structures, they don't have true nucleus, the bacteria don't have true nucleus and membrane-bound organelles. Fungi, they possess true nucleus and membrane-bound organelles. And for the cell wall, the main component of the cell wall of bacteria is peptidoglycan layer. So this also differentiates bacteria based on its gram-positivity. So like if it's gram-positive or gram-negative. Okay, of course, except Archaeobacteria. For fungi, they have chitin again. Next one, for the cell membrane, okay, the sterols are present in fungi. However, it's absent in Mycoplasma because Mycoplasma in the first place don't have cell wall. So Mycoplasma pneumonia, for example, Mycoplasma pneumoniae, that causes atypical or walking pneumonia. Okay, so they don't have cell wall, so they don't have sterols in their cell wall again. For mode of nutrition, they are heterotrophic, chemoautotrophic, photoautotrophic, aerobic, anaerobic, and facultative anaerobic. Okay, for fungi, they are heterotrophic. They can also be aerobic, majority of them, or facultative anaerobe. So when we say heterotroph or heterotrophic, meaning to say they are organisms that eat, okay, other plants or animals for energy and nutrients. Okay, but when we say chemoautotrophic, so these are organisms that obtain their energy from chemical reaction. But their source of carbon is the most oxidized form of carbon, which is what? Which is, of course, the carbon dioxide. How about photoautotroph or photoautotrophic microorganisms? So they are also called as photosynthetic organisms. So photosynthetic organisms like the green plant or cyanobacterium. So they can utilize energy from light to synthesize organic molecules. So that's photoautotrophs. Aerobic, of course, they require oxygen, okay, especially if they are obligate aerobes, so meaning to say they cannot grow in the absence of oxygen. But if they are anaerobic, okay, they are poisoned. That's the layman's term there. They are poisoned by oxygen. So usually, if you see a test tube or containing a culture medium, okay, anaerobic microorganisms grow at the bottom of the tube, whereas aerobic microorganisms grow at the surface of the culture medium in a tube. Okay, how about this one? Facultative anaerobe. So facultative anaerobes, meaning to say they are aerobes, but they can still live even in the absence of oxygen. Okay, that's facultative anaerobes. For the reproduction, bacteria or prokaryotes, they can produce or reproduce through binary fission. For fungi, either sexual again or asexual, mitosis or meiosis. And then another thing, the fungi exist in two forms. They can be yeast or they can be molds. So those are the general forms of fungi. So what is a yeast? So this is a single vegetative cell which is unicellular. And typically when you observe their colonies in a culture medium, they are smooth and the colonies are creamy. And then the morphologic appearance of all yeast are somewhat similar. So that's why when you observe them in the culture media, okay, the colonies look exactly the same or somewhat, again, similar to each other. And then the identification is based on the result of biochemical testing, and they reproduce by budding. That's the reproduction of yeast cells. So this is an example of a parent cell of this is a yeast. Okay, this is a mature cell. You can see here the nucleus, and then it will form a bud, and through mitosis, they can produce their daughter cells, which can separate from the parent cell. So we have here the general structure of the yeast. So this A, okay, this one, this is again the nucleus. Okay, this one, this is the bud. Okay, the bud, the dividing, okay, daughter cell coming from the parent cell, this one. Okay, letter C is the yeast, and letter D again, this is the nucleus of the yeast. So this one, this is the colony. These are the colonies, okay, of yeast. Okay, in particular, they can form these feet-like structures, okay, in the culture medium. So this one, this is an example of Candida albicans colonies growing on Columbia blood agar. So Candida albicans, okay, it's a yeast again, and a normal habitat of the human gut and of course mouth. However, it can be opportunistic. Okay, opportunistic oral and genital infections, especially in immunocompromised patients. Okay, and then also this one, this is an example again of a Candida albican, okay, or Candida albicans fungal culture on Shadler agar. Okay, so Candida albicans again, it's commonly found on the skin and the mucous membranes of the mouth. How about for the molds? So molds are fuzzy or woolly appearance, which is due to what structure? Mycelium. So as what you can see here, you have the mycelium. Okay, so what is a mycelium? It's the collection of long strands of root-like structures called the hyphae. Okay, mycelium is a collection of hyphae. So we have the term hyphae. So it's a thread-like chains of cells that form blank and blank. So that form the fruiting body, this one, and the mycelium. So this one, it's an underground network of hyphae that have grouped together. What is it again? That is this one, mycelium. Next one, reproductive structure of a fungus and grows above the ground. So what is it? Of course, the fruiting body. Okay? And we have here the hyphae, which is a fundamental unit of fungi. Next one, we have here the structure of the fruiting bodies, and then again, the mycelium. For the three types of mycelium, we have fertile, aerial, and vegetative or submerged hyphae. So for vegetative or submerged hyphae, these are those that penetrate the surface of the medium and absorbs nutrients. So they grow here. Okay? So penetrates the surface. So they are at the bottom of the medium. For the aerial mycelium, these are those that grow above the agar surface. So they are here, aerial mycelium. Next one, for fertile mycelium, they are aerial hyphae that bear reproductive structures such as the conidia or sporangia. So they are here. Okay, that a little point there, that's a fertile or reproductive hyphae. How about the two types of hyphae? So we have septate and aseptate. It's very obvious. The septate hyphae contains this septa or cross walls, whereas the aseptate, they don't have septa, they don't have cross walls. Okay, another thing, the types of hyphae based on their pigmentation. So if the hyphae is considered as a hyaline hyphae, meaning to say they don't possess any pigment on their cell wall, so they look transparent. But when the hyphae, okay, possesses melanin pigments in their cell wall, meaning to say they are pigmented, so we call it as phaeoid or demacious. Okay, so they have melanin pigments in their cell wall. So because they have that pigment, their colonies are colored gray, black, or olive. So examples of phaeoid or demacious hyphae, we have Bipolaris, Cladosporium, and Exophiala. Okay, phaeoid. So this is the colony of Bipolaris, this one the Cladosporium, and we have here the Exophiala. So consider also their colors, so they are pigmented. Okay, this is the microscopic appearance of Bipolaris. Again, note the dark pigmentation, which is again due to the presence of melanin in their cell wall. Next one, we have here the forms of vegetative hyphae. So we have the antler hyphae, so that's an irregular pattern. Racket hyphae, club-shaped hyphae. Spiral hyphae, of course, tight coils all throughout. Rhizoid hyphae, that's root-like. This is the most common, and it penetrates below the culture medium. Okay, so this is again the illustration of the different structures or appearance of the hyphae. For the spiral hyphae here, again, they are spirally coiled. Okay, and they are commonly seen or commonly observed in Trichophyton mentagrophytes. How about this one? I did not mention this earlier. Pectinate. So pectinate hyphae or pectinate body, these are short unilateral projections from the hyphae that resemble a broken comb. Okay, a broken comb, and they are commonly seen in Microsporum oduwuni. Okay, depending on how you pronounce it, that is how I pronounce it, Microsporum oduwini. Okay, next one, this is fabic chandelier. So these are the group of hyphal tips that collectively resemble a chandelier or the antlers of the deer. So they can occur in Trichophyton schoenleini and also Trichophyton violatium. So that's for the fabic or antler hyphae. How about for the nodular hyphae or nodular organ? So this is an enlargement of the mycelium that consists of closely twisted hyphae. So as what you can see here, you have here the enlargement like a nodule. Okay, so we can often observe this in Microsporum canis or oftentimes also in Trichophyton mentagrophytes. So nodular organ or nodular hyphae. How about this one? Racket hyphae. So these are regular enlargement. So as what you can see there, you have here 1, 2, and then another one. So regular enlargement of one end of each segment with the opposing end remaining thin. So it's like a thin and then enlargement, thin and then enlargement. So we can observe them in Epidermophyton flocosum. We can also observe them in Trichophyton, okay, Trichophyton mentagrophytes also, okay, just like in nodular organ. How about the racket hyphae? So again, racket hyphae, I've mentioned that already. One of the things that I have mentioned on the previous slide is the rhizoid. Again, what are those? Those are root-like structures, okay, root-like structures seen in portions of vegetative hyphae and in some members of zygomycetes. Okay, so these are the different structures again and appearance of hyphae. Okay, another one, so we have here the specialized structures, okay, that we can find again in vegetative hyphae or mycelia. This is a Rhizopus species, okay, showing rhizoids. So you can see here root-like structures. How about this one? Dimorphism. So dimorphism meaning to say the fungi have two morphologic states. It could be a yeast at 37 degrees Celsius and molds at room temperature. Like this Saborod's dextrose agar, okay, at 25 to 30 degrees Celsius, you can see, okay, in the microscope, this mold form. Okay, this brain heart infusion agar, on the other hand, stored at 35 to 37 degrees Celsius, can isolate a yeast form of fungi. Okay, so that is dimorphic fungi. They have two forms. So these are the examples of dimorphic fungi. So number one, we have Blastomyces dermatitidis. Okay, Blastomyces dermatitidis. Number two, we have Coccidioides immitis. So Coccidioides immitis, that is what we call as the valley fever. Okay, so they live in soil also because they are fungi. So people can get their spores, okay, can get valley fever by breathing in the spores of fungi. Okay, by getting the spores from air, of course, and usually if they get sick, they'll just get better, okay, within like weeks or months. However, there are immunocompromised patients that need antifungal medication when infected with this C. immitis. Okay? How about Histoplasma capsulatum? Okay, still inhalation of organisms from soil, but they are mostly associated with bird or bat droppings. Okay, of course, the bird and bat droppings that contain fungi. Okay, for Sporothrix schenki, this is also known as rose gardener's disease. Okay, so this affects the cutaneous area of the skin. Okay, and then also it can affect the lungs. So Sporothrix schenki. Okay, we will go there when we talk about the specifics of fungi. Okay, next one we have these structures. We have Blastomyces dermatitidis. Again, this one still is a fungi, so it can live in moist soil, okay, in decomposing organic matter. Okay, so still the people can be infected with Blastomyces dermatitidis by inhaling the mold that produces spores. Okay, so Blastomyces dermatitidis. How about for the reproduction? As what you can see here in the illustration, this is a giant puffball mushroom. So it bursts open, right? And it releases trillions of spores. And then, of course, because of this huge amount of spores, it can increase the likelihood of landing, okay, in an environment that will support their growth. Okay, so number one, we have asexual reproduction. For the asexual reproduction, this is the primary basis of their identification, okay, and of course the production of conidia through mitosis. So asexual mitosis. So conidiogenous cells. So we have here the structure. So take a look at it. So one of the examples of asexual reproduction is the production of a conidia. So we call this one as Phyaloconidia. So these Phyaloconidia or conidia are formed from conidiogenous cells. Like of course this one, the Phialide. So in this case, the conidiogenous cell is the Phialide. It's a vase-like structure, okay, that blow out, okay, the conidia. Okay, so meaning to say the Phyaloconidia are blown out of the Phialide. Okay, so again, these conidia are product of asexual reproduction. So Phialide again are vase-like structure. Okay, I'm repeating that one to make you easily remember it. So Phialide is a vase-like structure, but we have another structure. We call it as annelide. So that is a ring-like structure. Okay, ring-like structure, annelide. Okay, so these are Phialides, these are annelide. Okay, so take a look at the image A. So it produces or extrudes conidia. Okay, the tip here tapers. Okay, it lengthens and acquires a ring of cell wall material as each conidium is released. But here also, tapered and then, okay, it extend. This one, there's a ring-like structure here for annelides. Another one, we have the Arthroconidia. So the Arthroconidia is another form of asexual reproduction, like your Phyaloconidia. Diba? Your Phyaloconidia are blown out from the Phialides. But for the Arthroconidia, okay, again, asexual reproduction, it is formed by fragmentation of fertile hyphae. Take a look at this one. There are already spaces in between. So fragmentation of hyphae, it is forming Arthroconidia. That looks like this one. Okay, these are again asexual spores. Okay, so again, these are propagules which are formed by segmentation of existing hyphae. Okay, for sexual reproduction, what's the process? Okay, meiosis. So this sexual reproduction, okay, as opposed to the asexual, okay, involves two homogeneous hyphae. So as what you can see here, there is one hyphae, there's another hyphae, and the condition must be the two hyphae must be identical in nature. So in order for it to fuse and their nuclei, again, to fuse, they must be identical in nature. So this involves joining of two nucleus or two nuclei from compatible matching strains. So two identical hyphae, followed by meiosis. So at the end, you have there the production of zygospores. So that's the product of sexual reproduction. Two identical hyphae fusing together, the two nuclei fusing together, forming zygospore. So these are my references. So I hope this short discussion made you learn something today. Thank you.